Third, welcome everyone to our third webinar, and I'm super excited to have our very own Dr. Scott Kelley join us today. Dr. Kelley, interestingly enough, started off his career as a population geneticist working with beetles. And so he got his bachelor's degree at Cornell, and then he also received his PhD working on population genetics at University of Colorado. And interestingly enough, he's also had a brief career trajectory as an elementary school teacher before he started off on his career as a bioinformatician. He is, I think, by far the highest cited biologist in our department here at SDSU. And he also serves currently as the director of our bioinformatics graduate program. Dr. Kelley has published extensively in the field of metagenomics and 16S ribosomal RNA-based bioinformatics. And I'm super excited to welcome him and have him tell us a little bit about some recent stuff that they've been doing with network analyses. And so without much further ado, take it away, Scott.
Thank you. That's a very kind introduction. Yeah, I did a lot of population genetics. I would have called myself a phylogeneticist, but I did have a intersection between the population genetics and the tree. That was my whole idea, is to go from the population to the broad speciation. That was all a fascinating thing. And because I was so interested in phylogenetic trees, and the reason I got into beetles was because I was told that there are more beetles than everything else. And I thought there's got to be a lot of things to study if there's more beetles. I just wanted to know why there are so many insects, why there's so many beetles, and what drove that. And frankly, a lot of it is their interactions with plants. So flowering plants and beetle speciation go hand in hand. So that's a big part of it. But yeah, they were fascinating. And then someone came along and said, you know what? There's more bacteria in those beetles than there are beetles. And I was like, oh, there's something even more diverse than beetles. So I started studying microbes, and I kind of never looked back. It's easier to swab my desk for a bunch of microbes than go literally hack open a tree for a bunch of beetles. So and then I got into bioinformatics. So okay, so that's what I'm going to talk about today. And I just want to point out that this work is really the work of my graduate student, my master's student, Cheyenne, and he. Obviously we worked together on this, but all of the code was written by Cheyenne. So I just wanted to emphasize how much great work that he's done on this.
Do you want to share your screen, Scott? I
Oh,
don't think it's
it says I'm sharing.
hmm.
Let's try it again.
Let's try it again. Yeah.
Share.
There you go.
Okay, I don't know. It said share and then it said stop share. So, okay, so I'll be talking about our Python package called PyBootnet. And I won't try not to make it a dry, here's my code kind of talk. But we're going to talk about how it works, and also going to talk about why I needed it, but a lot about what networks are and why you should use it. So, and why we had to make our own package. All right, so let's go to the next page if I can get this to advance. All right, so first I'm going to talk about some background on networks. For those of you who aren't familiar with networks, this was also a good opportunity for me to teach networks to people who've never seen it before. So I made some slides for that. And I'm going to talk about co-occurrence correlation networks. You can also think of networks. A lot of network theory has to do with computers. So computers are networked together. That's what the net is, or the internet. And each computer is a node, and the connections are the cables between them. But a correlation network is like a network, but you put it together by whether things are seen together or not seen together. All right, so I'll give you an example of what I mean. So I'm going to give you an example of a network of dogs in a neighborhood park to show you how it works, and then we'll go on to the biological interpretations. So then I'm going to talk about measuring and interpreting networks and comparing networks from different conditions or environments and what you can learn from that. And then I'll give you some examples of molecular networks and ecological networks. So let's talk about networks. Let's talk about an example. I put this together to try to explain to people how co-occurrence networks work. And I thought, what if you wanted to know which dogs were interacting, and you want to make a networks of dogs in a neighborhood park, right? And so the data that you had was a bunch of observations or photographs of the dogs together, right? So you don't really have any behavioral data. You don't have anything else. You just have whether they're there at the same time or not. That's it. So here's an example. So here's 6 data samples that I took of dogs in a park. And so here's one thing you can find out is these 2 dogs are always seen together. So whenever one is there, you see the other one. All right. So be a correlation. So then the next one, here's another little corgi. I think it's a corgi that is also seen with these dogs. So whenever you see these dogs, you also see the corgi. But interestingly, the corgi is also seen with another dog. So there's this other dog that's always there with the corgi. I'm ignoring the other dogs for this point, just focusing on. So these are like co-occurrences. They occur at the same time, right? And then here's another example where in none of these pictures do you see this dog, the black one in the bottom right, with either of these 2 dogs. All right, so these are the kind of data that you could gather to put together a correlation or a network. And often when you do it with species, you would actually not do presence or absence, you would do the number of times you saw them, right? So if you see them, if you see more of one, you might see less of another. That's a negative correlation. If you see more of one and more of another, then that's a positive correlation. So as there are more of them, they're more, are they opposite or not? But in this case, it's just presence absence. So let's put together a network based on the dog observations. It might look something like this. Okay, so here's our network. And you can see I have the most important dogs, like the most frequent dogs in this network so far. So I have, for instance, I have a green, I'll go through it, but I just wanted to show about graph theory. It talks about you get a lot of terminology and I want you to go. So each dog is a node. This could be also like a computer node. That's where it probably comes from, but it's also known as a vertex in graph theory. I usually call it a node. And then the edges are the connections. And in this case, they're correlations, right? So in some networks, in a computer network, it would be just or a social network, this person is connected to this person. They have a link or a like. But in this case, what it means is that there's a correlation that when you see one, you see the other or you don't see the other one. So that's what an edge means in a co-occurrence network. All right, so let's look at what they mean. So a green line means it's a positive correlation. When you see one, you see the other. Or if it was bacteria, if you see more of one, you also see more of the other, right? The red line, the dash line or dash line is a negative correlation. So if you see one, you do not see the other one, right? So there's 2 examples where you don't see them. And that would mean that maybe in a correlation network, that usually means when you see more of one, you see less of the other. That's a negative correlation. Very simple. These are just pairwise correlations, by the way. And they're just like Spearman or Pearson. And then finally, you can actually look at line. There's other things like line thickness, which would indicate the strength of the correlation. So here's another example where I didn't see these other dogs very often. But some of them have correlations. They're just not as much correlation. So I don't have as much data. I didn't see this little Wiener Schnitzel or whatever it is very often, but it is correlated with this dog. I'll have a little example of that. And then this one is negatively correlated with this one and positive. So the thickness of the line can sometimes indicate, and not always, but sometimes they use it to indicate the strength of the correlation. All right. So network metrics. Now let's talk about how you might analyze these data, analyze this as a network and what properties the network was having. So these are often very important things. Like I'll show you some examples of that, but let's talk about that. So one of the things you might want to know is like, how many nodes does your network have? How big is the network? How many things are actually correlated? So this has 6 nodes or 6 dogs. Then you might ask how many edges there are. So here we have, I counted a total of 9 lines. That's 9 edges. And some networks have more and some networks have fewer. And then you can also ask which ones are positive and which ones are negative. This is going to come up again and again. So this one has 6 positives and 3 negative correlations. Then you also might talk about things like what are called degree centrality or betweenness centrality. And I'll tell you what I mean by that, because this is actually, a computer is also good for this, but let's look at degree centrality. So degree centrality, I might have this dog on the periphery here only has 1 connection, right? So its degree centrality is really low. It's not really central to the network. It's not important in some way. It's only one of them, right? Whereas this dog has 4 connections. So this is the highest degree centrality of the network. And this is important because, for instance, a computer that had a high degree centrality would be a very important computer for the network. If it goes down, it could take the whole network with it. In a biological system, it could be in a very important species that everyone that influences all the others. Like it could be like, in the forest, it could be a fig tree in a rainforest that has all these other plants that it's associated with, and it's really a key species. In this case, it could be something else, and we'll talk about that. There's also betweenness centrality. Betweenness centrality means how connected, closely connected it is to everyone else. So how many routes through this node to get to everyone. So this one basically is connected to everything by just one or maybe two. It's actually almost only one, which so gives it a high betweenness centrality, whereas this one has a low betweenness centrality because it takes a long time to get to say you have to do 1, 2, 3 paths to get to this one. And so that means, sometimes those are metrics that are used to judge a network. I'm mostly going to focus on edges and nodes in this talk, but I wanted you to know that these things exist and they're important for understanding ecological systems, or they can be. They can lead to more hypotheses. Okay, so now let's interpret the network. This is very important. I wanted to put this in because here's what we have. So what does a positive correlation imply about our network? Well, we could talk about that, but a lot of people think that a positive correlation means that they're directly interacting. So one is feeding the other, or they work together somehow, or they're good friends, or something like that. Another one is a negative correlation, means that they basically are against each other. There's competition going on, they fight each other, then one poisons the other. This is the exciting part of the network. Most people are focused on that, all right? But we'll talk about that. What about a highly connected node? That one could be central to the graph. Like everything goes through that node, that if you take away that node, like it could be like taking away sharks in an ecosystem, the whole ecosystem falls apart because sharks eat everything. Who knows? But I do want to point out that you can overinterpret these numbers. So for instance, if I asked in this network why this dog had all these positive correlations, well, it could be it's a friendly dog, right? It could be like it loves all the other dogs and it does it, or it could just be that it's always there because it lives in the neighborhood, right? And that's where the correlation and causation thing come out. You shouldn't confuse those two things, right? And I'll talk about that in other slides, but basically you have to be very careful in your interpretation. It could be that it's interacting with all those dogs, or could be it just lives next to the park, right? We don't know. And you can't tell from this data. You'd have to watch its behavior to know what it actually did. Here's another one. Do these dogs hate each other? Like, do they fight all the time? Or do their owners come at different times of the day, right? And then this one, these two dogs, do they come because they interact with each other, or do they have the same owner, right? So those are questions that you would have on a network because to make sure. So correlation does not equal causation, very important. But I will say, if you do not have a correlation between two of them, then you don't really have a hypothesis to test. So what I can ask, for instance, I now have a hypothesis that this is an important dog and that it's friendlier. I can go and test that. But it might just be that it's just there because it eats it likes to be there, it's next to the park. So that's the same with bacteria. For instance, I might say, if these were bacteria, I might say, hey, these are interacting in a positive way. They're feeding each other, or they're just growing on the same substrate. So they grow at the same time, and that's why they're correlating. All right, so that's it. Okay, so the other thing I want to talk about quickly is comparing networks. What type of co-occurrence networks do we see in different neighborhoods? So this is part of what our software is doing, is not only looking at network metrics, but asking how different are two networks under different conditions. So let's look at a dog network again. I'm going to stick with this for a little longer. But let's say we had two neighborhoods, a neighborhood A, a neighborhood. We did the same correlation networks for both of them, and we got these two very different looking networks. Why are they different? Well, there's several reasons they're different. This one on the left has more positive correlations than the one on the right, right? It also has higher connectivity. So Leo here is connected to everybody, sort of like the previous graph, and there's more interconnections among them. This one over here has huge amounts of negative correlations and only a few positive correlations between pairs, right? So why could this be? Well, one of them is a dog park. So they're always interacting. And there's some dogs that are friendlier than others, or they're more often. This one could be an apartment complex. And you would see that you only saw them together in the lobby, right? So the only time you ever saw them was when they're going out together. Totally different kind of network. So what I'm saying is we may not understand. If I looked at this, I can't tell you the differences, why they're different. But I can tell you there's something totally different about these networks that you can go and explore. And this is why networks can be really useful. for predicting interactions and for also predicting and understanding communities. Okay, so here's an example that's more biological. Here's a soil ecosystem. So these are bacteria in a soil. And so this is something we're asking questions of what happens in a natural soil environment versus a disturbed agricultural environment. So one thing you might expect is in a natural soil environment over time, they become to be more interrelated and they feed on each other and they cooperate more. Whereas in a disturbed environment, there might be just like whoever can show up and they always fighting, right? Because they're trying to get, it's disturbed and it's just new species that can come in and colonize and just like a weed, they want to fight everyone else, right? So those are very, very different networks for the same kind of ecosystem. All right. Here's a couple. Now that you've seen this, this is when people throw a biological network at you. You're like, oh my God, this is horrible. But now that you have some understanding of how they're structured, you can look at this and say, oh, I kind of understand. Because everyone honestly shows their networks in a different way. So let's look at the network on the left here. This is an mRNA expression network. And it took me a while to figure out what they're doing. But what they have here, this is pretty weird, actually. The middle one is the triangle's transcription factors that regulate gene expression. And there's the other ones are genes that are getting regulated. And so, for instance, over in the left here, top left, I hope you can see my mouse, there's one that's correlated with 3 genes. If it's orange, the correlation is positive, which means if that transcription is found, that gene is is higher expression. And the green is if it's negatively correlated. So this one over the left is highly correlated with one and negatively correlated with the other, which means that maybe it regulates them. Or it could be just coincidence. It could be like when that's on, it's become some other pathway. The two pathways are on at the same time. I don't know the difference, but I could knock this gene out and ask if it affects transcription. That's a cool question. Here's another example on the right, which is more like mine. These are immune factors in genes, and these are actually people's weight and height and triglycerides. So they're actually doing correlations, positive and negative correlations within and between people's like weights and heights and triglycerides and also the immune factors that they're doing. You can see that a lot of the immune factors are correlated, which makes a lot of sense, highly correlated, because that's all part of the same immune response. And some of them are correlated with, say, adiposity, or you can see that there's negative correlations within these groups and that kind of thing. So that's 2 reasons. Here's another thing that I really like this network. It looks crazy, but you'll see what I'm talking about. This is microbial networks from the gut of a chicken. So this is chicken gut microbiome. And what they did was they had 206 poor little chickens that they killed so you could eat. And then yeah, I know. Well, you guys eat chicken, I mean, at least they're doing some science. So what they did was they took microbial communities from all parts of the digestive tract, duodenum, jejunum, ileum, cecum, not all of it, but little sections. And for each of them, they did a microbial network. And I thought it was really interesting for a couple of reasons. First, you can see these weird architectures there. So notice that they drew theirs a little differently. They have all this, I think there's phylum up here, and they have all these interconnections. And I think the gray ones are positive correlations and the green ones are negative. This is how I drew it. But you can see that they have different sizes. They have different numbers of connections. They have different architectures. Like what is this little outside one in the top right in duodenum? I don't know. And they have different, like down in the ileum, they have a bunch of species that are hugely negative correlated with basically everything else. Like, why is that? That's totally cool. And here in the feces, there's like 2 completely different sets of networks. Like you have one and you do not have the other one. So maybe this is 2 states of the microbiome. All very cool looking networks. All right. So the goal of the project, now that I've told you about it, is to create a way to analyze these networks. And we created PyBootNet, which was Python bootstrap networking. That's what it stands for. And it's to create a flexible, expandable, and easily maintained Python software package for analyzing networks, especially statistically comparing networks from different conditions, but it does all sorts of things. And so I'm going to walk you through how that works. All right, so there was a similar package that we actually tried to use in R, which exists. So maybe you can get it to run. We were not able to get it installed, which is why we wanted to. But it does some similar things. It bootstraps your data, it constructs networks, but it doesn't directly compare to the network as far as we can tell, but we couldn't get it to run. And one of the problems is that it has 21 dependencies. And it actually has 8 optional dependencies. So it's not really surprising that it would crash if people can't pay to maintain it. So it's written in R too, and R is great, but Python is more, in my opinion, it's a better integrated programming language. R is great for data analysis, but if you wanted to scale it up massively in parallel, it would be hard. So that's another reason to do it. And it just didn't have all the features we wanted, as far as we can tell. Again, we tried for weeks to get this thing to run. All right, so we wrote our own package to do some other things. And what we really were interested in is you could make a network, but how good is it? Like, how confident are you that it's correct? And so that's where the bootstrap statistical test comes in. So what we were doing is we have one set of data, but we want to know how robust it is to perturbation. Like, is this correlation real, or is it based on just like two things that are seen together one time, and so one way to do that, and this is a very common technique for lots of data like this, is a bootstrap technique, which is a random sampling with replacement. And I'll show you what I mean by that. But if you can randomly resample it over and over and over again, and you keep getting the same result and keep getting the same correlation, that's strong evidence that your data support that correlation. All right, and that's what we're going to do. So how does that work? Okay, so this is an example of a bootstrap replicate, and I'll walk you through it, of a single data set of samples and features. So over here on the left, I have a data set of bacteria. So bacteria 1, these are 6 bacterial species. And that's on the top. That's the rows and the columns and the rows of the samples. So these are like the pictures of the dogs. So sample 1 would be one picture of the dogs, but it's a bacteria. And it's the same idea, except instead of just presence and absence, I'm counting how many of each times I see them. And I do this based on sequence data. So I'm doing sequence counts. And then I have sample 2, 3, 4. So I can make a network on the original feature table. No problem. But I'm like, well, how good is it? So one way to ask that question is to bootstrap it over and over and over again to see if you keep getting the same results. So here's 2 different bootstrap replicates, just random resampling, where I randomly resampled the samples. So I go in and I say, give me the same number of samples. That's 1, 2, 3, 6, but randomly sample with replace. So I went and grabbed 4, and then I grabbed 1, and then I grabbed 1 again, and then I had 3, 4, 4, and I stop when the data set's the same size. Then I make a network, right? And then I do it again. Sample, 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 and I make it. And then I do this 500 or 1,000 times, and I ask what are the correlations for each network, right? So I'm asking, like, do networks for all of them, and then I can do something like this. So here's the network that I got, all told, from all of these, of all the species. And I didn't quite do, I missed one here, but anyway, doesn't matter. What I ask is, what are the average correlations between correlation coefficients from all of the 500 or 1,000 bootstraps that I did, right? And so if this one on average was 0.9. So that means that there was, a very high correlation between bacteria 1 and bacteria 2. There's a 0.74 between 1 and 3, et cetera, et cetera. And then what I can do is to say, I'm only going to accept them if they're above a certain threshold. So after all my bootstrap replicates, I can get rid of them. So I got rid of, like this, I say my threshold in this case was, if you can see it, it's 0.8. Right, R equals 0.8. So when I'm done, I can just get rid of the anything below 0.8. And then when I'm done, this is my final well-supported, statistically well-supported, at least by the bootstrap network that I'm left with. So these ones I can trust more. That's really the idea. Okay, so the good news is we found out that you can do it for more than the networks. You can do it for every network statistic, not just whether there's correlations, but we can actually calculate for each network, we can actually calculate the total number of edges, the total number of nodes, the overall between-us centrality, the density, the transitivity, and we can even do a statistical test. So every time we do a network, we do all the networks within it, and then we take the network and we say which are the best ones. Then of that, we say how many edges do they have, positive or negative, how many nodes, what's the between-us centrality. And we have 1,000 values for every one of those. All right, so this is how it works in practice. We do each replicate, we create a correlation matrix, we build a network, then we calculate the network metrics. And we do it for replicate 2, replicate 3, all the way to the total number of replicates at the end. And I just wanted to show this is the PyBootnet GitHub that Cheyenne worked on. So it's looking pretty good. We're going to update it for the publication. I think we're going to get that accepted. Very exciting. And right now we have 20 different functions in it. Things like, bootstrap replicates, correlation matrices. So they're all, what I really like about the way Cheyenne put it together is all of these say exactly what they do. So you know exactly how each of them work. There's 20 different functions, and there are more to come, including statistical tests. All right, so now I'm going to show you, now that I've created, I'm going to show you how it works and how it can analyze some real data. So we did 2 different data sets that are very, very different from one another. One of them is from a built environment, and I'll show you what that is, but that means an indoor environment. So we were looking at microbial growth on surfaces and building materials. Mostly they got wet, because when you ever get a building surface wet, anything, you know that mold grows on it. You know that bacteria grows on it and starts to decay. So we were interested in how those networks form and can we make predictions about them. And the other one was a gut microbial network and a hypogonadal mouse model. So I also do work in gut microbes. And I was looking at, with my colleague over at UCSD, we're looking at the influence of puberty on the gut microbiome and how that would affect the microbes in the gut and also how it would affect the networks that are formed. So those are 2 very different studies, and I'll step you through them. All right, so the built environment data is a nice place to start because it's pretty straightforward. So what we did was we took a bunch of building materials and we cut them into little coupons, and we had lots of replicates, which is always great. Replication is great. And we had 3 different kinds. We had oriented strand board, which is more like plywood. We had gypsum board, which is drywall, and the gypsum is not something they grow on, but there's a paper on it that if you get that wet, mold will grow all over it. And then medium density fiberboard, which is like this glued thing. These are very common building materials. And we had already shown that there's a huge, so what we do is we put them out in the room and we let stuff fall on them. And we just let them inoculate for a month. And then we got them wet. So if we don't get them wet, we knew that nothing is growing. It's just whatever lands there. But if you get it wet, things go nuts. Bacteria and fungi, and only certain kinds. So that was a previous study, but this study, what we did was like, well, what happens if they dry out, periodically? So we had 2 treatments. We had a wet treatment where they always stayed wet for the whole period of 49 days. And then we had a drying cycle every week where we dried them completely out. We wanted to see what happened to drying, because you can imagine a situation where you periodically wet the materials, it gets dried out, it gets wet again, that kind of thing. All right, so those were our studies. What we did, and this is for people who care about this kind of stuff, is we took our data and we did what's called a centered log ratio. We had a feature table of all the species like before, but we did a centered log ratio transformation of these data because our data is compositional, and this deals with compositionality. So just so for those of you who are interested, and it keeps you from having a lot of false negative correlations, which are very common in relative abundance data. And then we had conditions. We had 2 conditions, material type and also the conditions, wet or cycling, right? So the map columns was used to transfer everything. Looks really pretty. All right, so let's talk about how we use the thing. So here's an example of a couple of functions that Cheyenne came up with where we can tell you how many bootstrap replicates do. So we're doing 500 bootstrap replicates. And what is our cutoff for correlation values of 0.8? You can change this. It's really easy. In some cases, we actually did have to change it because we weren't seeing anything. But most of the time, 0.8 was really good. And that's a pretty high correlation threshold. So 0.8 is not bad for biology. So what we did was we did 500 bootstrap replicates of the wet condition in this case. And we did a Spearman correlation on each of the replicates. We drew the networks, cut off the correlations, and then we calculated 5 different network statistics. So those network statistics can be seen over in the right here. So for first replicate, this is what we got. And the replicate number 1, we calculated the number of edges for that network, the number of nodes, the average use centrality, the transitivity, blah, 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 blah. We do this for every network. For the second replicate, number of edges, number of nodes, and then 5 network statistics. So we can do it all at once on the networks, very convenient. Then you can take that data and you can take all 500 replicates and you can plot it. So now we're starting to see some interesting stuff here. All right, so here in this case, what we did was we have two different networks, one based on the bacteria and one based on the fungi, right? And you can see them automatically, they're the exact opposite. So what are we plotting here? We're taking the mean number of edges, which means the total number of correlations of all the materials that have been wet. We put them all together. We didn't separate them by material type in this analysis. We put them all together. And all the materials that were in the cycling, right? And then we said, what is the mean of all of those things? And you can see that they're completely different. And then we can also see from the wet and the cycling as well, for the fungi species, it went the opposite direction. So the wet had higher numbers of correlations than the cycling. So the total network size, networks and things that are correlated, which overall different between wet and cycling when you put them all together. Right. Then we did a different thing. This is the correlation of the fungi, but what we did was we did wet and cycling within material type. So we have three different material types: OSB, oriented strand board, wet and oriented strand board cycling, MDF, wet and cycling, and gypsum, wet and cycling, which is really the drywall. And you can see that, first off, there's very big differences between the material types. So we had the highest one was MDF for the number of edges. Oriental strand board was medium, and the gypsum was lower, right? And then we know that we have a statistical tests, which I will get to, but we know that they're really quite different after all those replicates. Which was really interesting, though, what I noticed is that so we saw a big difference between wet and cycling, right? I just said that, but not within materials. So there's a contradiction here that I was like, I'm trying to understand, but if you combine them, there's a huge difference between wet and cycling, but if you leave them separate, there's no difference. We didn't see any statistical difference between them. And this is the same in the bacteria. And I think I know why that is, but it's certainly something that's curious about the two systems, right? And I have to think about it, but I thought that was really interesting. Okay, so then the other thing that really popped out for us, now these are some of the networks they're building. So here's a network from wet boards, the medium density fiberboard, and wet boards and gypsum. And you don't need to know anything about networks to know these are completely different. Over on the left here, these are all species here, and these are they're all positive interactions. There's not a negative one in sight, and they're all like really, really tightly correlated around a few species here. In the gypsum, we have something where we see a bunch of species, fungal species, that seem to love each other. They all grow on the same thing, but they're completely negatively correlated with all these other species. So whenever you see these guys, you do not really see these other species. So it's like, even though the network statistics weren't that different, the networks themselves are totally different. And I don't know why this would be the case. All I can think of is there's some species up here that are fighting, that maybe outcompete with the other ones, right? So if you have these, you just don't have these other ones. And that doesn't happen on the medium density fiberboard. So we'll have to look at what species they are to figure that out. All right. And then I wanted to go into, sorry, I'm starting to lose my voice a bit. I wanted to go into the statistical tests that we can do, which I was really proud of. I didn't come up with this test, but I adapted it from a website, which was kind of fun. And so what we have here is, for each bootstrap replicate, I'm going to show you the different networks. These aren't actually the networks. I just threw in some examples to show you. But here's an MDF network, and here's a gypsum network. And then the second bootstrap replicate, I have an MDF network and a gypsum network. So it's the same networks, but I'm randomly resampling them each time. And you can see that in the first one, if I do a pairwise comparison, I see that edges are 140 and there's only 90. And you can see that in the graph, right? There's fewer edges in this one in the right than the left. There's also fewer nodes. So I can do these statistics for all of them, and I can take data for all the bootstrap replicates. So here's some of my data. And so every time I'm doing a pairwise analysis, I say how many, I'm just doing nodes right now, or edges. So edges, 140, 90, step replica 1, 151 to 95, step 2, 137 to 100, 122 to 87, right? So what I'm saying is every time, in this case, the higher one is given a 1 and the lower one is given a 0. So I just change it to 0, 1. This is the same as saying heads and tails. All right, so basically I flip four heads in a row, right? So every doesn't matter what I do, MDF is, at least these four samples, is always higher. If I did 500 replicates and every single time MDF was greater than the GW in the number of edges, this is like flipping a coin 500 times and getting head every single time, which means that they're not the same distribution. They're totally different. And so that's the binomial test we do. And that's why I just want to explain that these p-values are really, really, really small. And that's because these networks are really different. There's nothing similar about them. Every metric, every single time you bootstrap them, they always come out different. And that's like a 500 heads in a row most of the time, right? So at least in some examples, we got these extreme p-values. So that's our statistical test. But it isn't always like this. Sometimes it wasn't like this. And I'll show you examples where they aren't different. Okay, so what is my time like here?
You're doing good. It's like 12:37, so
12:37. Okay,
yep.
so good. I can make it through this one. This I only have a couple slides here, but I just want to go through the project. It's a really cool project. It's a little complicated, but I'm going to make it as simple as possible. So this is from the paper we have, and I want you to point out on the left side, this is a mouse model that has a mutation so that the mice do not produce steroid hormones. That means that they never go through puberty. So what this is we've knocked out the gonadotropin releasing hormone. And if it's a truncated version of the gene in the model, and this means that the mutant mice do not go through, they do not produce proper GnRH, they do not produce LH and FSH, they do not go to the ovaries and testes or produce estrogen or testosterone. So both males and females do not go through puberty. So the bottom line is do not make steroids, do not go through puberty. And the question we had was, how does this affect the microbiome? So over here on the right, we had groups of mice that we followed over time. And I'm only going to show you week 10, but we followed them prepubertal, peripubertal in the middle, and adult. So they go through puberty really fast and they start getting into estrus. But notice, so we have a group of wild types, these go through puberty, and a group of mutants that do not go through puberty. And I'm going to show you the week 10 data based on the 16S RNA bacterial data. All right. Hopefully this all made sense, but we're really asking, like, we got a pretty good sample size here. A lot of mouse studies are done with 6 mice. So I'm very proud of what we have here. And the question is, well, how does this, we know it affects the species, but how does it affect the networks? So let's look at the networks of these guys. All right, so I'm just going to show you one stat because it was one that I can pop up. And you can notice that, so we have here the mean number of edges per group at week 10 post-puberty. So this is at week 10 after they've gone through puberty. What did they look like? And this is after, I think it's 500 bootstrap replicates, or it might have been 1,000, I can't remember, but it's close to that. So what we see here is we really don't see any difference in the number of edges, which means the total correlation, although it was slightly smaller with the wild, but there was no statistical difference here. But we did see a very big statistical difference between the female wild type and the female mutants. So the mutants had way more correlations on average than the female wild type, right? And if you look at this graph, it gets, I think, a bit more interesting. So here we have the 4 networks that we visualized. And immediately your eye can go to the bottom left, and you can see it's got a lot fewer lines. It's just got fewer lines, right? And you bootstrap it, it's very different. So the female wild type, and look at the female mutant, tons of lines, lots and lots of correlations. Very cool. And the male wild type and the male mutant are a little different, but it's not different. It's different. So we had 4 groups at week 10, numbers of edges and nodes. Everything increased over time except for the female wild type, which I thought was very interesting. We have positive correlations. We also had these clusters, which were just like that other sample. When we have this cluster, we don't have this, which means we have 2 states of the microbiome. We either this group or this group. And that's true of everyone. Oh yeah, that's the negative correlation. So there's lots of positive correlations and lots of negative correlations here, which means they're sort of not, when you have one, you don't have the other. And then only one clear group. There's only one real cluster in the female wild type. We saw a little bit, but it wasn't like the other ones where there was these strong correlations. And I think I know why this is, this is my hypothesis, is that Females are going through estrus, which is basically the mouse's menstrual cycle. And what I think is happening is if there's big changes during the estrus cycle in the gut microbiome, right? So the other ones, they don't go through that. So the bacteria are basically always there. And if they're always there, they're always going to be correlated, right? They're always going to be available. But if you start messing with it, suddenly you have some are there and some are not. So you're going through estrus and you're changing the gut microbiome, the bacteria changing, those correlations are going to disappear because they're not always there all the time. So I think of the reason these are always here is because the system isn't changing. So they're always seen together because they're always there, right? But when you throw in estrus, it changes the system. That's my working hypothesis. And then the last thing I'll show you is like you can actually investigate with this thing. What are the most interesting nodes? What are the most connected nodes in the system? And so for here I can rank them. And this is what Cheyenne did, but I can rank the top most connected nodes, the ones that might be the most important in the system. And then I can look at how different they are between the wild type, and there's some species I don't even know what they are, and then the mutant, which has a whole different set of species. So, and we were actually able to look at what those clusters were as well. Like what were those clusters all about? They weren't the same species, but they often were the same families. So that was really cool. Okay, so in conclusion, PyBootNet allows us to do all this stuff. It's easy to work with. It's easy to maintain. I can understand it. And if I can understand it, anyone can understand it. And it can cover complex relationships and biological systems, and it's Python code, so it can integrate pretty easily. In the future, we need to do better version control and some pip install, better tutorials and vignettes for people. We're also looking at new functions for comparing different bacteria. So I want to compare bacteria versus fungi. In fact, we just started doing that, and that's the most interesting correlations because the fungi control bacteria. So when you see certain fungi, bacteria are totally gone, probably because they're producing antibiotics. That's cool. But we could also do like genes versus metabolites, and we want to parallelize it. And, we want to, I don't know what the last thing is. Oh yeah, we want to test the correlations within the network to see if we see the same bugs correlated all the time as well. All right, so with that, my acknowledgements, obviously Cheyenne is the top one to acknowledge, and I hope he's on the call. But Dr. Miller, Luke Miller, and Cristal Zuniga. Luke especially helped us with the bootstrap and understanding the statistics. It was really helpful. And the Kelley Lab members for doing this. And I would like to thank NSF for no science funding. Okay, I finally got funded by NSF after 20 years, but they denied me for decades, despite me being a lot of publications. I couldn't squeeze any money out of those guys. So. But finally, we got some money, so I can't say that joke anymore.
We're still waiting on ours too, so.
Yeah, but you've been funded at least. I went 0 and 12. 0 and 12 when I started. I just gave up. I was like, these guys want too much. So anyway, but you're a better scientist. You're at least a better grant writer. I must be a terrible grant writer.
No. Great stuff, Scott. I'm gonna probably, I don't know, have your students sent you questions? There's a handful of ones that people have been putting on the chat window.
Oh, right. I have to go to I usually control this because I go to the website.
Do
all
let me
this.
ask you a few that are on here in the meantime. So I had a question actually. Let me start. So I'm curious when you do the bootstrapping with, if you have multiple samples, which are basically your rows and your columns are your different species, and your question is to see if the species abundance or species network is actually, significantly meaningful. Wouldn't you be bootstrapping the species as against the samples?
You want to keep so we talked about that. And I think the problem is that we wanted to keep, I think it's the samples you want to bootstrap because you want to keep the same species in the network at all times. So if you bootstrap the species, I mean, you can do that, but I think we wanted to keep the numbers, the species together when we were bootstrapping it.
Okay.
We thought about it both ways, and I can't remember exactly why, but we thought this is the way to go, is to keep the it's sort of similar to the well, I'm trying to think. It's a little bit similar to phylogenetics where you keep the column, so you keep all of the nucleotides.
Right.
Anyway,
Columns would be
we had a
nucleotides,
reason for
their
this.
samples. Yeah, yeah, yeah.
Yeah.
Cool. Yeah, no, that makes sense. Okay, so let me read the next one was how do you decide on the number of bootstrap replicates? Why 500?
Yeah, that's a great question.
That's from Alice. Yep.
Yeah, no, that's a great question. So the rule of thumb is something like 5,000 or 10,000. In practice, it pretty much stabilizes after a few numbers. I do think that there is an issue that I would like to do more, but all right, sometimes on a laptop, Cheyenne could not get it to finish, so we went with fewer. But in practice, we found that after like 500, we weren't getting much of a change. So it's not a really exciting, like they say 5,000 or 10,000, but I would say it also is somewhat dependent on the number of samples because there's only so many, if you only had 6 samples, for instance, there's only so many combinations of those you can even pick. But we did find as our sample sizes grow, as stuff got more, as the matrix got more sparse, it got harder and harder to create the networks and calculate all the statistics. So yes, we probably underdid some of them. Yeah, but yeah.
Yeah, I almost always feel like it's an arbitrary number. The more the better, right? But then it gets computationally intractable, and then you're at some point overfitting. So
Yeah, exactly. Yeah,
yeah.
exactly.
Lindsay asks, what type of dependencies does the package require, and why does it need those specific dependencies to work? I'm curious, in your case, do you use like NumPy or something to do some of the math?
Yeah, we use NumPy to do the mathematics and I think the vector stuff. I mean, I didn't write the code. I'm sorry. I should know more. We use network X, which is to draw the networks. And I think there was one or two more. I forget. If Cheyenne's on it, he could write it in there. But yeah, there were sort of the basic ones that we really needed. And I think with the exception of network X, although I think that's pretty well documented matplotlib, I think. Matplotlib. Yeah, so these are very well-known libraries that are updated frequently, and I don't think they're going to have a problem crashing. It could be the network X does every now and then. We have a little bit of issue with that sometimes. But yeah, just the bare minimum pretty much.
Hina asks, what website did you obtain or update your binomial statistical test knowledge from?
Oh,
For yeah,
it's funny. I was just looking around like, how can you, I couldn't find anyone who was like, they had a real test to do like compared to now. They had a couple of them and I just Googled around and I finally found somebody who did a test about, I think it was people walking over 2 bridges. So they would take data about whether one bridge was more trafficked than another bridge. And they would say, okay, day one, how many people did you see on bridge one versus bridge two? Day two, bridge one, bridge two, day three, bridge one. And then they had a little test that they said, well, it's just like heads or tails. And I was like, oh, that's a binomial. That's just head flipping. So I was like, well, that sounds like what we could do with our networks because each one we just have a number. And sort of like people going across the bridge. And I thought, well, if it's one, if it's higher in one, then it's a one. And if it's always higher in one or more higher in one than you expect by chance, that's like flipping a lot of heads more than you would expect. So I was just Googling around. I was really trying hard to find someone who did this kind of thing. Mostly bootstraps are done within these data sets, not between. So
yeah,
there's lots of stuff within data sets to like, what's the R value? What's the like population genetics? You guys use that a lot.
yeah,
Phylogenetics, so you do it for the branch links, but it's like you're not really comparing 2 trees in that same way. So
yeah.
I was like, this is pretty simple.
Yeah, and I've been super interested in this as well because we're trying to approach a population genetics problem by modeling sort of network structure across the genome.
Mhm.
And network structure here is population structure across the genome. And I've sort of come across at least a couple of strategies to where people use, if you're comparing, let's say, population structure network in one individual versus another individual or a group of individuals. One way that people approach it is to sort of just represent the network itself as a matrix, right? Which is basically your distances and your weights on your nodes, um, and calculate a variance covariance matrix from the distance matrix. And then what that gives you then is you have a matrix structure for each network, and then you can just do a Mantel test of correlation between the two. So
Oh
it gives,
yeah, yeah, yeah. I had thought about that. I
yeah.
just realized, everyone, I'm so sorry to my students that I was so busy putting my talk together that I didn't give them a chance to put in questions.
Oh, but there's a lot of questions on here, so we get lots of
Okay,
them.
so I'm just telling my students now that I have updated, let me see, I'm updated the Google spreadsheet. And so you can get extra credit. I'm so sorry about that. And I'm just going to put in, yeah. So now you can go to the Google spreadsheet and you can type your questions in and get credit for it. Sorry about that.
And the other one that I've commonly seen is where you calculate the for each one of those statistics. Let's say network, what is it? Connectivity, or what were the other some of the other ones that you mentioned?
Connectivity, density,
Density,
betweenness, centrality,
yes,
you
right.
know,
So you have a
yeah.
distribution for all of those, right? And
Right.
so what you can do then is to just take the cumulative density distribution or CDF of that distribution, and then you'll have a CDF for network one and CDF for network two. So then you can do a Kolmogorov-Smirnov test between the two,
Ah,
which just looks at the CDF. And it's kind of, it's basically what you are doing with your binomial test, except you are doing that for individual outcomes. Instead, if you're looking at overall network structure comparison, the CDF seems to work, the Kolmogorov-Smirnov test.
and
We
is
can
that
talk.
only pairwise?
Yeah. What's that?
Is that only pairwise?
It is only pairwise, but I feel like there is a multi version of that.
Because one of the things I think that's a really great that you mentioned that, because if you had, say, three distributions,
Yeah.
you could theoretically ask how much overlap of the three distributions there are,
Yep.
and you could do multiple tests at the same time. So that's a really great suggestion because I was looking for a way. Right now, we're just doing it in pairs, which
Yeah.
is fine. But I like the idea of doing multiple at the same time and asking, are they similar? Are
Are
they
they similar?
almost like
Yeah.
an ANOVA or something?
Yeah, I think there is a multiple comparison version of the test. I'll have to
Oh,
look it up. I'll tell you.
okay, thank you. Yeah, if you could
Yeah,
send me
no,
that, that would be fantastic. I was looking for stuff like that, and I just I
yeah,
couldn't think of it. That's a great idea. That's an easy, probably really easy thing to
it
implement too.
is. Yeah, and you can write it in Python or R. It's super
Yeah,
easy.
exactly. SciPy or something like that. Yeah.
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 So then Justin asks, how do you determine an appropriate number of nodes to include in a network?
The nodes that you include are, in our case, are based on the ones that are highly correlated. So we basically do the networks and we do it over and over again, and we only include nodes that have at least one significant correlation with somebody else. So if it doesn't have, if it's a node that isn't correlated with anything else, which happens a lot, it's just like you only saw it once. It does not get included. It obviously immediately gets thrown out because it's just like it just has no correlation with anything. So that's how it works.
Uh, awesome, thank you. And then we have Melissa who asks, why was one of the female mice labeled 17 versus the others all labeled 20? Is there a possibility to integrate another coding language with the program?
That number indicated the number of mice in the study. And so for all groups except one, we had 20 mice. And the reason we only had 17 in the females is that you actually have to, these are all mutants. And the mutants, when we first did it, they were like, I think the females were eating the mutant. The moms are eating the mutant females. So for a while there was like something going on we don't understand. And then we only had ended up 17 with those, which is, as much as it was hard to get that many of the mutants because whenever you get the mutants, you also get wild type. And to get the right number of mutants, it took months and months and months to get that data. So it was a lot of work. Yes, and I think this would be easy to integrate with other programming, like other Python languages and maybe anything. Basically, the input data is just a table. Like if you have a table of features and samples, you can run it. We didn't tell you what your feature should look like. We didn't make it CLR transformed, anything. You could have a protein network. Someone asked about how you do protein networks. I would just have all my proteins, all the samples, and all the counts of how many proteins I saw. That's it. It's just an observation network. Most of our data is just observation networks, as far as I can tell. That's
Yeah,
like 90% of our data is a two-dimensional table of numbers, you know?
yeah, I can see this. I mean, as you were talking, I can easily see this extensible to what we're doing with population structure because it's literally just a two by two, right?
Yeah.
I mean, yeah.
Yeah, and
Uh,
you don't have to CLR. You could just use, if you have abundance data, you just throw it in because
yep.
it's agnostic. But we knew our data were compositional, so we changed it.
Yeah. And so to as a follow-up to that then, so for your data sets, I'm assuming you obtain count tables from 16S or ITS sequencing
Yeah,
uh, from these, right? So generated through CHIME.
yeah. But we could do. We also had metagenomic data. So
Oh,
what
you
we did with metagenomic data was we assembled species, and then we used the reads to count the species, and that's how we got the count tables. And then we had metabolomics data,
did.
where we just had like the peak area peaks of all the metabolites, and that was our feature table. And in the fungi, we used a different gene. So you could, any table like you could work with that you think is, independently derived samples. But yeah, you could do this with RNA expression data. All it is like RNA, here's all your genes, here's all your counts, how do they interact, and then you could say, how are the networks different? And were the strong correlations or not?
Yeah, and I have a grad student who's looking at protein-protein interaction networks
Yeah,
and so
we
network
can do that as
structure at
well.
large, right? Which is the same logic again, right? You can construct those interaction networks based off of anything.
Yeah, I'd love
Um,
to work with you. And there's other questions like, well, a lot of people are really interested in the individual correlations of gene X to gene Y and whether it's in the same in different networks.
yep.
And there's a jackknife approach that I want to do that because that's a within network analysis, which
I was gonna actually ask you, like, because once you have the engine for doing bootstrapping, adding a jackknife should be relatively straightforward.
trivial. Yeah, it's trivial. That's the next stage. Yeah. Yeah.
Um, Awesome. We had one more question, and I'm watching the time as well, just to be conscientious. It says, at any point, if any, in the data analysis or just testing, did you assume a normality of the data?
So the nice thing about the bootstrap, as I understand it, is that it's kind of it doesn't assume a distribution. So it creates it basically, because it's sampling within the data, it doesn't have to these random sampling don't have to assume a normal distribution to work. I do think, like you said, the data probably end up being normally distributed. So my guess is that after you do all the bootstraps, you get these, you're going to get these beautiful, perfect little curves because of just the way it works. But it's possible you have a different distribution. The good news is that you don't have to worry about that with the bootstrap because you can still get a mean and standard deviation from that and ask if those are different regardless of how it's actually distributed. That's a great question though.
Yeah. Yeah, but if you sample enough, I bet you it'll be a normal distribution regardless.
If after 500 or 1,000, I would bet money.
Yeah. Well, because that's what you're doing. You're just doing sampling with replacement,
I
right? So
know.
you're bound to end up with a normal
Exactly. Yeah.
distribution of whatever network structure that you're doing. Well, it's 1 o'clock. Are there any other questions from the audience? Scott, this was awesome. I think maybe one in here. Oh yeah, here's one in the Q&A. One more. Hannah asks, what would you recommend in terms of best practices for publishing network graphs? How can we make it easy for other people to interpret specific interactions from what we're publishing? I imagine it would be difficult to track individual relationships between nodes across different publications. How is this relationship between bacteria changing between different environments? Maybe some interactive visualizations, text representations of the graph. That's a
This,
great
this is like
question.
a thesis.
Yeah, I know.
These are amazing questions, I have to say. There's so many of them. I don't know if I could answer them all. There is a real inconsistency of graph and network strategies of posting. I've seen all sorts of things. I've seen the circle ones. I've seen all that. Best practices. I mean, I do think there's a lack of statistical rigor. So you don't know if these things are real. So that's what we're hoping to do is make sure and making them interpretable for other people. Like that one graph looked really ugly. It's like, why would you have a line to a positive and a line to a negative? I think that's bad practice. But make them visually so you can see where are the positives, where are the negatives, where are the nodes, and then definitely make sure that you do not overinterpret these things, right? So I think a lot of the positive correlations of my graphs are simply like on the building material. You could say, oh, these guys are growing together, they love each other, they're feeding each other. Or you can say, well, here's a substrate, and they both eat that substrate, right? That's a correlation. So don't assume that they're actually interacting, but assume that the correlation is real. That's like the ice cream and what is it? There's a strong correlation between ice cream sales and murder. It's like, yeah, they are correlated, but they have nothing to do with one another. You know? There's some other good questions. I can't remember. Maybe one more. I don't know.
So it was about like using like interactive visualizations of
Yeah, I
um,
mean, I think one of the things you could do is,
graph
you can
structures.
look at the networks and you can say, like we were doing, what's the most connected node? What is its interaction with everyone else? And then, think hard about why that might be. But the next thing you should do is to say, well, I really think these two things are interacting. You have to manipulate it experimentally. And you have to say, okay, these two things are constantly doing this. I think this is my hypothesis. They're definitely correlated, but does one actually affect the other? You have to probably it impacts one of them and see how it affects the outcome, right? Or just repeat it. Now, the other thing is, I did see this other question of like, well, I don't see the same bugs each time. That is a
Mhm.
big thing in the ecology system. And I think what we should look at is, do they have the same function in the environment? So you could have two or three different species that are, in each one they look completely different in terms of species, but actually they're the same family, or they have the same functional genes. So those are things that I might look into. And the last thing I'd say is look for other things that might be interacting. So we looked at bacteria networks, but we found that the fungi might be more important affecting the bacteria than actually other bacteria. And if we haven't done the co-network, we would not know the answer at all.
Mhm.
So I don't know if that helps, but those are some things I've come across. Great
All
question.
right.
Excellent
Yeah,
question. Everyone's
awesome
got some great
questions.
stuff.
Um, All right. Well, it's 103. So thank you, Scott, for doing this. This was awesome. Next week, we're going to be having one of Scott's former students who's also on this call, I think, which is super exciting because this time around, we actually wanted to invite some alums from our programs and talk about what they've been up to for their bioinformatics research after graduation. So we're excited to have Dr. Jennifer Fokir join us. And so yeah, we'll meet back up again next Friday at noon. But for now, thank you so much. If you have any questions, please feel free to email us. So, all right.
Thank
Thanks a
you.
lot.
Thank you for being such a great host.
Thanks, Scott. Take care.